Okay, so good afternoon, everyone. My name is Marta Teperek, and I'm from TU Delft Library. I'm working in Research Data Support Services, and we would like to welcome you today to our Future Forward seminar. Today we will be discussing FAIR data in microscopy. So why would you make microscopy data FAIR as findable, accessible, reusable, and interoperable? What's that? How would we do that? What can we learn from the experience of our colleagues who will be introducing this topic to us? And just to let you know that if you would like to tweet today at today's event, we have a hashtag dedicated for today's seminar series, so that would be nice to see you online as well. And just to give you a little bit of background to this seminar series, that's a 5th seminar in this series, and we really want to talk about bringing science forward, about opening up our research results and sharing with the world. But we do really realize that the topic is perhaps not yet commonly accepted. There are people who perhaps are in disagreement with open science principles. There are people who have difficulties in making their data fair or open, and these are all valid reasons for discussion. So the aim of this Future Forward seminar series is really to facilitate discussion, to gather in the same room various people who are big proponents of open science, but also those who are perhaps not sure how to do that, or perhaps have some very valid reasons for not making their data or their research openly available. So really everyone is welcome. The main purpose of this series is to stimulate discussion. And before we start, I would like to ask you a question, and if you can do a show of hands, I would like to know if you have attended any of the previous seminar series in the past. So if I can ask you to raise your hand if that's the first seminar that you're attending. Okay, about half I would say. And if you can also raise your hand if that's one of the series, so if that's not the first one, if you've attended already some of the previous seminars. Okay, I would say half-half, which is great, because we always would like to welcome some new people, of course, on top of the people who enjoyed the previous seminars and continue attending those. Thank you so much. And in terms of some practical notes, we will have video recording. Today you can see the camera and you can see our colleague Jos from Collège Rama doing the recording. The purpose of this is to also be able to share the video and the experience with colleagues who were not able to attend the seminar. And I have to say, as a praise to our speakers today, there was lots of interest from researchers from all over the world who wanted to attend the seminar. They found out about it via Twitter, but couldn't because of distance, because of the cost of travel, but would really like to see the video of your talk. So that's why we always record our seminars and make them available afterwards. But what it means also, we'll be recording the question and answer session. So if you don't like to be present in the video, please speak to Jos, who will be able to make sure that you are not part of the video recording. And another important issue, if you will be speaking, asking a question or answering a question, please make sure that you introduce yourself and that you also use the microphone so we can hear you clear also on the video. We can all hear each other clearly in this small room, but on the video recording that might not be so clear. And the final reminder, my colleague Santosh down there is guarding the entrance, and he has feedback forms for you. So you can see that's these nice cards where you can tier your feedback about the event. And he also has the attendee list. So if you have entered the room, you haven't signed on the attendee list, or you haven't grabbed that feedback form, please do so and give it to us before you leave so we can continue to improve those seminar series in 2020. And just to finish up with the plan for today, we've already did the registration. I'm just doing the welcome remark. We will have the talk by Katie and Bernd very soon. We will then have closing remark by the head of Department of Imaging Physics at TU Delft, who is also the line manager of Bernd, one of our today's speakers today. And this will be followed by Q&A session, so you will have the opportunity to ask questions of the speakers. And we will then take you to the drinks reception. And without any further ado, I would like to introduce Esther Plomb, who is the data steward at the Faculty of Applied Sciences, and who will introduce today's speakers.
Thank you. So it's my great pleasure to announce our speakers of today. So Kati Wolstencroft is an assistant professor at the Leiden Institute of Advanced Computer Science, also known as LIACS, and she leads the Semantic Systems Bioinformatics group. Sorry, I had to look that up. She studied biochemistry and bioinformatics at the Universities of Manchester and Leeds. And she is a founding member of the FAIRDOM Foundation, which is a UE platform to share FAIR data in systems and synthetic biology. So it's very exciting. And she's also one of the authors of the FAIR Guiding Principles for Scientific Data Management and Stewardship, which is better known as that one paper from 2016 that everyone cites when they're talking about FAIR. Yeah. And Bernd Rieger has done his studies in Germany at the Technical University of München and did his PhD here at Delft in imaging processing and analysis. He worked as a postdoc at the Max Planck Institute for Institute for Biophysical Chemistry in Göttingen, and worked for FEI, Electron Optics, here in Eindhoven. He's currently a full professor here at the Faculty of Applied Sciences in the Department of Imaging Physics, and he's in the field of computational microscopy, combining imaging physics and processing for light and electron microscopy. So that explains the title of today's talk. So Bernd is going to start us off, and then Kati will follow with her presentation. So without further ado, I would like to invite you to the floor.
Thanks for the introduction. Okay, we wait a bit for the presentation. Thanks a lot. Okay, try not to stand in front of the screen. So today will be about microscopy data and how to make that fair and why will we do that. So what do we cover in this next half hour about? So first I want to introduce you a bit to microscopy data. Now what is that? What makes it maybe different from other data? So who of you has used a microscope? Okay. Who uses it on a regular basis? Okay, so there's a few. So I give you a bit of an overview what this data is about and how does it look like and what could be particularities of this data compared to maybe other data that you want to store or to access. Then of course the question, why do we have this talk? Why is this not already common sense? Why don't we do that? Didn't we not do that 10 years ago, but why now? And then once we have discussed this, then Katie will tell you how we're going to do this. So if you have a question or comment, please raise your hands. With such a small group, you can always make an interruption. That's fine. Okay. So first, so what do we use the microscope for? I mean, microscope data is not acquired because you like to have pretty pictures, but typically you want to discover something. Now, what do you want to discover with microscopic data? That's microscopic stuff. Now, what is that microscopic things that you want to discover? So we often use the picture that has the orders of magnitude, and it starts, let's say, with small animals in the order of a few nanometers, and that goes down to, let's say, atoms that you see here. So what can you see with light microscopes? So with light microscopes, you can see on the micrometer scale. So that would be on this order, 10 to the minus 6 meters, 1 micrometers, or 1,000 nanometers. That's the typical scale that you could see with a microscope. Now what is that typically? That would be on the order of cells in your body. They would have typically that scale, but they would be a lot smaller than, for example, human hair. So we investigate a lot biological samples, and that's, you see here, for example, a cell, but that's also how microscopy was introduced. Van Leeuwenhoek, who started here in Delft, he was the first to look at cells, bacteria, something on the micrometer scale. So what I will talk today about is light microscopy data. But of course, the same type of data images will also be acquired in electron microscopy that can be used typically for smaller things. So that would be more on the order that's a bit smaller, maybe on the nanometer scale, the size of the DNA, for example. But the type, so the problems that I will talk about, they will be the same for light microscopy and electron microscopy. So I want to give you some examples of things that we're looking at, and also why that might not be a problem to store that data or to work on that data and to manipulate this data. So one thing on the largest things that you can image, that would be zebrafish with a microscope now. An adult zebrafish is on the order of a few millimeters, on a few centimeters. That may be like this. And you have them in the natural form. Here they're called wild type, and you see these stripes that you might be familiar with from aquarium. But they also exist in a mutated form where they're nearly transparent. So that is very good, because then you can use the microscope to look through it. Otherwise you don't see anything, you only see the skin. But if you want to look inside. Now what can you do with that? If you have a look at an embryo, so the embryo would only be on the size of a few millimeters. You see here down, you see an embryo, and then this embryo is stained with the fluorescent markers, and these different green and red, that would be fluorescent marker that you would see through the fish because it's made transparent. That's things that you can see with a microscope. So that's on the largest scale that we typically image, would be small animals. Okay, so this down. What happens if we go down in scale when we want to image? So then you can, if you go down from animals, then you could go to tissues, and then you go from the tissues you go down to mammalian cells, and then maybe bacteria, and then subcellular parts. So mitochondria, that would be the machineries, the factories inside your cell that produce all the energy. They will be smaller than the cells, and there would be a subcellular compartment. Now, with light microscope, what you typically cannot observe is something smaller than about half the wavelength of the light, and that's the resolution limit that is given by the diffraction of the light that you use. And all these smaller things that you see here, that they're not impossible to observe, but then you have to use cool tricks to make that work, or you have to use an electron microscope. Okay. Well, you will also see something that extends this region a bit. That will be called super resolution microscopy data that we also store. Now, another type of imaging that's used a lot in light microscopy is what's called high content, high throughput microscopy. So here you see cells. For example, this blue thing that you here see, that would be the cell nucleus of a cell, and then you see the outside of the cells, and you see many cells, or here you see also many cells. Now, this high content screening, what you do, you image many cells to discover, for example, the working of drugs. So you have untreated and you have treated cells, and you see now, does it make a difference? Now, the only way to test it or to see if that makes a difference or not means you need the statistics. Now, and you don't have to can use one drug, but you have to use a lot of drugs. So that means you image a lot of cells, a lot of different conditions. And you want to study what's the effect on the cells. So for example, do they live, what changes their morphology, and so forth. So you take a lot of data, and that's automated. Now you already see that this will produce probably a lot of data because you do an automation. This automation can run the whole day, that's not a problem. Also in the night. And then you have to analyze all this data and find back what you have done. So that's already one step smaller. Now if you go to the smallest scale, what you can image is subcellular compartments. And for this is, here you have two examples of what's called super resolution microscopy. Here you see part of a neuron, so that's a cell in your brain. So this is, here you see these neurons, they make connections to each other. And what you see here, that's a three-dimensional recording. And so what you see is a sort of a lattice-like structure that forms a cylinder around this of here, these labeled proteins, a cylinder around this. Oh, that's not going too well. A cylinder, and here you see it from a side view, around this, these connections that you made. And the size of that cylinder, that's very small. It's only 200 nanometers. So that's 0.2 micrometers. So that's a lot smaller than what typically bacteria would be. That was published a few years ago. Also wanted to show you something from our lab. So this was done together with Sjoerd here. This is the nuclear pore complex, what you see here, imaged in 3D. This nuclear pore complex is a hole in the nucleus of each of your cells. And so that means all protein that's needed. So inside the cell nucleus is all the DNA, but you have to get the information from the DNA out, and then all stuff happens. It has to go to the very small pores. And these pores is something that you see here. So we have acquired them in 3D. There's also an animation that hopefully... so this is this pore that you see, and then all the things that would go through that channel like this. Now the diameter of this pore is only on the order of 100 nanometer, so that's a lot smaller than what you could typically image with a light microscope. But it's people pushing the envelope. Now that's typically these three examples that I've shown you here. That would be typical data that people in microscopy acquire. That also worked with the beamer. Okay, perfect. So the question is, so why do we want to have this fair data now? So there, of course, there's not only one answer, but a lot of them. So one of the things is that a few of these technical advances and the things that have been invented, which I will show you in the future, in a minute, they have opened new areas, and people work a lot more on this light microscope to use more things. Then the other thing is that automation of the microscopes now make it possible to operate them for 24/7. So that means more data is generated. That means also typically you don't just look at the image and say, okay, I now understand the process, or maybe on a live acquisition, but you record many times the same thing to get statistical information. Now, and that boils down to the third, so that this part here, because of the increased computation power that you have, and the way images are acquired, you don't look at them, but you also have to process them a lot. So now if you process them, that means some sort of black box is going over the data, and then things happen or might not happen, and you have to know what is that. So maybe you want to be able to find this back. What is that data? When was it acquired? How was it done? And maybe the biggest thing is that because of these things here, you acquire now much more data than you did maybe 10 or 20 years ago. So that's a big driver. So I would say this is the science case. So why scientists think it's a good idea that also we would benefit from a way to make data findable and that other people can reuse it and they can check that. Because if somebody acquired lots of data, why cannot somebody else use that data? That only works if you know what you're doing. So now we'll show you a few what are the reason of the technical advances in the field that have made that happen. One is this super resolution imaging at the smallest scale, the Nobel Prize a few years back, that made that possible. So now the field is maybe 13 years old, and that really made a big impact in that sense that a lot of people acquire new images. You will see that back. Another thing is called, it's a sheet of light. That was an invention by this gentleman here that you see here on the side, Ernst Stelzer. So here the idea, so here you see again a sketch of a zebrafish, and this is how you typically illuminate a fish. With the blue light you illuminate the fish, then the fluorescent from the fish comes back and you record the green light, and this is what is captured on the camera. He said, nah, maybe that's not a good idea. Maybe we should illuminate the fish from the side, here with this blue light, and then record whatever comes back here. Now that was a breakthrough in that sense, because this blue light is very damaging to the fish. If you now want to image the fish, or something that's relatively large and living, I mean, you can experience this yourself. If you sit outside in the sun, your skin will get red. That's okay. But what happens to the fish? Because typically, so this light dose that's put on here is typically on the order of 10,000 suns. So now you can understand why typically a fish or a cell would not survive more than a few minutes. Now that's not nice. That's good in the terms of data, because in a few minutes you cannot acquire a lot of data. But if you now this idea that because now a lot less light damages the fish, you can image the fish or small animals, larvae, embryo, for days. Now that means you also acquire data for days. So that's nice. And then in the field of electron imaging, a few years, 2 years back only, the Nobel Prize was given to three gentlemen, plus the invention of this camera that you see here. That's a much more sensitive camera than you had before. This together, that also in this field, the data that you acquire increased a lot, because the cameras became larger, more sensitive, faster, and you had the benefit of automatization. And then, to give you a bit of feeling for now, you know what the pictures look like, so how much data is it really that you acquire? So the largest data that we typically acquire come from this light sheet microscopy. So one acquisition is typically a three-dimensional image plus something in time. So you acquire a volume and then a movie of this volume, because you take multiple images of the fish during development or while it lives. Now, what is the size of the camera? The size of the camera is about 2000 by 2000 pixels. Then you acquire 100 stacks to make a volumetric image. Then you acquire this maybe over 1 hour. Now, if you do all the computation, that means one acquisition of this fish is on the order of 1 terabyte. Now, so that's a lot of data to store, but also to process, and also just get from the microscope camera to your hard drive. So that's the worst thing that we could do in terms of acquiring data. The other data that we can get is high throughput data. That's typically not as bad, because you only have on an order of, let's say, a few million camera frames with the same size. So data could be on the order of maybe 10 gigabyte per acquisition. And then the super resolution imaging. So that's also not acquired from one image, but from a very long image stack. But the image stack is not 1 million, but maybe on the order of 10 to 100,000 frames. So that's also a bit smaller in terms of size. But still, all these acquired data, that's a lot. If you have that, sometimes you can make very nice things. So here, for example, I'll show you one of these light sheet recordings. Now this is on the order of, on a few days, you see a root plant growing. So they imaged the roots of a plant in a watery environment, and really you can see apparently the imaging does not hamper the plant, it just grows and grows and grows. And it is, you see the cell walls and how it goes. Now that's very nice for developmental biology, but you acquire a lot of data. So here's my slide on what typically metadata looks like of the recording. So that's not a joke, typically there's nothing. So why, then you can ask yourself now, why is that? That's in the beginning, often these techniques come from self-built hardware, or people use something that's not a commercialized system. So that means people would write the metadata in their lab book, but not store that together with the raw files. Sometimes you have people think, okay, that's a problem, the metadata, so they have some home-built solution. But that only persists as long as the PhD student is in the lab, and then it disappears. Now, if you buy a commercial system, there are 4 large vendors in the world. Of course, these vendors, they have metadata that's already acquired from the microscope, but that will not tell you what you image. That only tells you, okay, so this, the laser power and percentage and the wavelength and the objective and all these things would be stored, but you don't know what you're really looking at or what is the conclusion. And what's also very bad, so all these 4 don't have a common file format, so each vendor has his own one, and that's often proprietary, so there's no way you can access it. And of course, they change this every few years. So that means the exchange of data to open software, that's really cumbersome, because typically that works by re-engineering the proprietary file format. That's not nice. So the data retrieval. So once you have acquired the data and you have stored it, how would the retrieval typically look like? So mostly, because university research is organized along projects, and the projects are financed by PhD and postdocs. Now that means often that the data is also really unusable once the PhD student or postdoc leaves that has acquired the data. If at all you can find it then. Because if the student leaves, you might also even not know where it is, because you don't have a centralized archive. Somebody puts it in a home disk or on a group disk, and that might or not get erased or taken somewhere else. So how long does the data really stay with you? A few years, I would typically say. Now, the data analysis, that's also a big chunk of the, let's say, the finding of the problem nowadays, because you acquire so much data, and the data is very large in 3D or in time often. You need automated analysis. And the data, so if it's 1 terabyte, that also does not fit in the memory of the computer. And even loading parts of that takes a long time. So what that implies is, where typically before in a biology lab, they would look at the data and have a conclusion. Here there's some sort of image analysis step in between before you can get a conclusion. Now that also, because of these problems mentioned here, that often requires some sort of specialist that's not a biologist. So they can either buy software to do that, but if you're on the forefront of research, you have to maybe collaborate with somebody. So the typical best task, for example, to transfer data is to put them on a hard disk and then go by train to the other person. That's often, if you have a large data set, that's the fastest way to do it. Some other problem is that the computation should not be done on a supercomputer, but should of course be done in the lab where they do the real experimenting. Otherwise they again have to wait so long. So now coming back to why now? Maybe not from a science perspective, but from other. So who else is pushing in this FAIR data? That's the reproducibility of science. And there have been, can you reproduce an experiment in general, or with microscopy in particular? But also there's been frauds in journals. So people, and why was that? Maybe because they could not access the data. Also, you see that microscopies now become expensive. So what happens? They will not be in each lab individually, but people buy centralized facilities where they use that. So that means to be a facility manager, and he's also expected to store the data, to organize the data, maybe to help you with the image analysis. So these people, they say, okay, now that's very nice that this is my job, but I mean, is there a solution somewhere out there to help me with this? And of course the funding agencies, because they give the researchers the money, and if you cannot reproduce the dark science, so that was what was the point to do this in the first place. Another thing that you might ask yourself, so is this common for all research fields? No, probably not. So there are examples in, especially in astronomy, where there's a long tradition to share data and also to store it in a better way. Now why is that? Because acquisition in astronomy is very, very expensive. So I cannot have just my own satellite and acquire data and then say, okay, the other group, yeah, okay, I don't give you my data because it was only €1 billion. So they have to share it. And also, so that they really, but also other fields like bioinformatics where maybe data acquisition is not so expensive. It's very common that people would acquire data and they would, in a standardized way, make this data accessible to other people so that people that, for example, work in the field of bioinformatics always work on data from other people. That's very uncommon in the imaging field. So I would say we really are way behind on how the community operates. So this is a more, let's say, a societal case compared to the science case where the science people, the researchers themselves directly benefit. Now, why are these big things? Here's a survey from Nature that they ask a bit more than 1,000 researchers, and they said, now, is there a reproducibility crisis in science? And now let's say 52 said a significant crisis, so that's a lot. Only a very small majority said there's no crisis, so that's not a good thing. Now, and how was that based? So they asked, for example, have you failed to reproduce an experiment? So then they split this out in different categories. So they said, okay, did you fail to somebody else's experiment and yourself? So if you a few years later try the same experiment again. Now, so here's the 50% line. So that's not a small number that people could not reproduce their experiments. So the question is, of course, why? I mean, so they ask this why. It is a really long list, but what you think it's not bad luck. So that people could ask. So what they think is from this survey is the most prominent problem is selective reporting. So somebody reports a finding, but let's say that's your typical best result. So somebody does a cherry picking that they find an effect once, but not in the 99 other cases that they did not report. So you don't just know that, you have to look very, very long to find it. So also the pressure to publish, but also low statistical power. So that has really nothing to do with bad luck. So this was a few years ago. So things have not been solved because I also found that last year there still was exactly the same problem and that they tried to solve that. So then of course is the question, so why is this so bad? I mean, in science would be something that you should be able to reproduce, and if you make something wrong, then they say, okay, you make it wrong, and then after some while we agree on how it should be done. So here's an example from our own research that 2 years ago I published together with Sjoerd. So there we took the time to check somebody else's result. And so now the problem was, in order to get this thoroughly checked and to make the experiment nice, this takes about 1 year of a PhD project. So 1 year, that means also the funding of 1 year next to the time from the supervision. That's a lot of money. And so these people did not even, did not intentionally do something wrong. But maybe it could also be the first two problems that they had here in the list. So they found something, but maybe they did not really understand it too good, so they just published it. So this is the reason. So the problem is, do people really care? I mean, do you care enough to spend 1 year to make this a case? But of course you can talk to somebody else. Have you seen this research? It does not really work. We agree, and then it dies. That also happens. I want to show you one example that was published in the proceeding of the National Academy of Science a few years back. So people had a look at that, and they just, everybody directly thought, okay, that's a scam, that cannot be true. But, so there's really an agreement in the community that this is really very selective reporting. But nobody that I spoke to, that everybody said this is nonsense, took the time to show that. And this is a high impact journal. And because people did not feel to do it. Of course, that's not nice. So what was here the problem? Was the underlying data available? No. So here you see an algorithm operated on some images, and then they compare the results. Now, so what you would like is that they give you the data and they give you the algorithm. And then you can test it yourself. And you can change the parameters or whatever. That was not the case. And that's not 20 years ago. That's 2013. So if you have access to that, that would be a lot easier. So also I would say in the imaging field, the journals and the funding agencies, they take the lead in that, because that's very bad for a journal, because this gives a journal bad repetition. So now journals started to require that you provide raw images or the data with the results. Yes?
Just a question. So we say that journals require this. Is this the editors, i.e. other researchers, who are enforcing or promoting that this sort of requirement be made?
So from the journals that I know, that could be probably on the editorial board, they decide that they want this because of the problems with the fraud. And then they say, okay, if you want to publish with us, please provide the data. Some journals now have reporting summaries where you say, okay, I have investigated 100 cells and I found this in 5, and you must disclose this. So they explicitly ask you all these questions, and if you would sign it, and then it really is a fraud. Before you could come away just by not telling. So there's a bit of a, depends a bit. So for example, for code or for an image analysis on the data, some, for example, must provide code that could be executable, so you don't know what's inside, or must be open source. So there are different degrees on what they require, but this now really comes through, and I think the journal pushed this. And they also provide, this costs them because they must provide facilities to store all that stuff alongside with your publication. And especially in case of raw data, there can be quite a bit. Okay, so what do we gain from doing this? Why do most researchers think that's a good idea to go through this process? One large part of the image analysis chain nowadays is artificial intelligence. So these are learning systems, and these systems can only learn by example. So you need a lot of examples. Now, how do you acquire a lot of examples if everybody shares their data? That's one part. You can also, of course, compute on somebody else's data. That's nice. But also, if this system would be there, you can also refine your own data or somebody else's data, because you can more easily compare something. When I started my PhD, then it was not so strange if somebody published an algorithm that there would be no code. That means if you want to redo it, that costs you 2 months to re-implement an algorithm. 2 months. So nobody would do this anymore. They would just say, okay, why it's not on GitHub? So we don't have a look at that. So to make impact, so that other people use your stuff, you have to be, they should be reproducible, and people should believe you that this works nice. Now, how that does help if you make this accessible? And with that, I want to stop and give Katie the floor.
Oh, thank you. Okay, thank you. So, there are lots of reasons why we should be making microscopy data fair, and there's lots of reasons why it should be now, and I'm going to now talk about how we plan to do that. But first of all, to add to all of the points that Bernd just made, we can all agree that there are some very good reasons for doing this. We can all agree, I think in this room, because we're all here, we must be interested in open science and fair data anyway, that these principles are very good ideas in general. But that's not normally enough for an individual scientist to actually put lots of effort into making their data fair. Because the person who benefits from all of the hard work of making the data fair is not necessarily the scientist who does that. It's other people who get to reuse that data. So still we have to deal with the fact that all of this extra annotation, all of this data management that we have to do in order to get there is seen as an extra additional piece of this puzzle which is not part of the science, it's something you do on the end. So that's still there. So for any initiative that's going to work, we have to ask, what are the real incentives for the individual scientists? What do they get out of following any recommendations for making fair data? Now, as you heard in the introduction, I've been working in fair data for a while. I've been involved in the production of the FAIRDOM platform, which is about sharing data, fair data in systems and synthetic biology. So FAIRDOM stands for Fair Data Operations and Models. And in systems biology, you have basically two sets of researchers working together. You have a group of people who work on mathematical modeling of biological systems and a group of people who work on investigating those same biological systems in the lab. And they come together to combine their ideas and their research into a cycle of modeling, simulation, new experiments. Experiments inform new parts of the model, and so on and so forth. So one of the largest incentives for doing any kind of fair data sharing for systems biology is that by sharing with your colleagues, the people that you have to collaborate with, it makes the whole process easier in the first place. So there was a big incentive there just from that point of view. The other thing that we observed was that because systems biology was an interdisciplinary subject, you would generate lots of different types of data, transcriptomics, proteomics, metabolomics, and when you came to publish, all of those data types would go into various different silos of data repositories, and the mathematical models would be somewhere else over here. So the only place where your whole experiment was represented was in your scientific publication, which is not computationally amenable. It's good for people reading it, but it doesn't help people understand and reuse the data very well. So that was the other big incentive for the systems biology community to get behind this idea of the FAIRDOM Hub, because it was a place where the whole experiment was represented and all of the different components of the experiments, the data, the models, the samples, the protocols, how those things fitted together, oops, let me go back, was all there for everybody else to see. And so by simply understanding that, we got an awful lot more buy-in from the community. And of course, the platform itself allows not only sharing of data, but also all sorts of processing of data. So you can do simulations of models through the platform. You can link out to other databases and pull more data into the platform. We now also have a link to Galaxy Workflow, so you can do various kinds of bioinformatics analyses on your data too. So all of these things came together to allow the individual scientists to see the advantages of this and the incentives for them. And so that's where we start when we think about making microscopy data fair. What are the incentives for the individual as well as for society and for science in general? So we follow the same rules. We don't want to reinvent anything. If something already exists, we build on top of it. We fit in as much as possible with what scientists are already doing. If there are standards already, we build on top of them. If there are common practices already, we build on top of those. We respect the requirements for the individual, we respect the requirements for collaboration groups, and we make sure that the scientists remain in control of their data. So that's what we did with the FAIRDOM platform. That worked, so we want to do the same again for NL-Bioimaging. So, what are the incentives for an individual scientist? Well, Bernd already outlined the fact that actually once you have your image, then there's probably an awful lot of work to do on processing and analyzing the data before you can actually understand what those images are telling you. And if you have an environment where you can do the acquisition, the annotation, and the analysis all together, then that is an incentive for the individual, especially if you're not yourself an expert in machine learning or an expert in these advanced analytics techniques. If there are tools and resources that you can tap into that the rest of the community has built. And there are some tools already that we can just reuse. We don't need to reinvent. There's a whole suite of tools built around the open microscopy environment. And this is it's a global initiative led by Jason Svedlow in Dundee, and they have a database which has been particularly designed for bioimaging, and they have a common exchange format, the OME format, and they have a series of plugins that allow you to go from various different proprietary formats that are produced by individual microscope companies to this common exchange format. So it allows you to pull in all sorts of different information into a database that's been specifically designed for bioimaging. So this is where we start. There's no point us trying to develop a new database for bioimaging. This is already in the community and being used in a lot of places. So this is what the OMERO database looks like. This is part of the Leiden OMERO instance that we have. So you can see when you ingest images, then it automatically gives you little thumbnails of those images. So as a human, you can go through and find the images that you're looking for. It also automatically ingests all of the metadata that that is produced by the microscopes. Well, these are the established microscopes, so these are the ones where the metadata formats are already well established. And then you can add all sorts of metadata yourself to those experiments. So you can tag things, which is shown over here. You can tag the images. You can tag what you've done to those images. You can say things about the experiment as a whole. You can describe the experimental setup. You can describe what's happened to your samples before you do the imaging. But this is all quite free-form at the moment. You can add what you like. Some people add lots, some people don't. So of course that's something that we need to try and encourage people to do more of, and we need to standardize how people do that. And then the OME standard data format allows, well, basically everything to work together. So you can go from raw images to processed data to data management, analysis and visualization. And within the platform you can actually plug into all sorts of different kinds of analysis tools. So this is very similar in my mind to what we have in the FAIRDOM platform. So this was very good news for me starting in bioimaging for the first time to try and replicate what we've done in systems biology. The other thing that happened reasonably recently, a couple of years ago, is that the OMERO database was chosen for the main public data repository for bioimaging, and this is the IDR. This is just a screenshot for that. So it's based on the same technology, and it's only been going for about a year, but it's already got 142 terabytes of data in there from the community, and that's 1 million different experiments. So people are willing to share this data. They are doing in large amounts now. So we just have to make that process easier for individual scientists. And then together with the IDR, we have the Bioimaging Archive, which is hosted by the EBI. And that also, well, the IDR is for kind of high quality, well-curated data sets that people think will be well reused. And the Bioimaging Archive is essentially for everything else. There's not a really good definition for what should go in the Bioimaging Archive, but that's the general idea. So, what are we going to do in NL Bioimaging? Well, this is the general setup. If we say OMERO and the OME is the basis for everything, then we have various different NL bioimaging nodes around the country, all doing different kinds of microscopy, and each node has its own OMERO database. Excuse me. And then on top of that, we have a national node, which is hosted by Subcelluar, because that's their core business, that's what they're really for. And they also have the added advantage that they've developed something called the Science Collaboration Zone, which is an authentication system to allow people from different institutes to share resources and to log into various different systems. So we can plug into existing infrastructure there. Now, above the national node, we have the Bioimaging Archive, as I just described, and the IDR, which is split into cell and tissue. Excuse me. And these are actually part of the Euro Bioimaging umbrella. So there's another organization that sits above the national initiative, which is European-wide, that we also combine and fall into. So this is the grand vision. Now, we can't do this until the NL Bioimaging proposal is fully funded, but what we can do in the meantime is show that this is actually a sensible approach. And so that's what we're working on now. So in Leiden, we have the Cell Observatory. Whoops, wrong button. And the Cell Observatory serves multiple different institutes across the university with many, many different microscopes. And we have now an OMERO instance set up there, which is connected to 4 different microscopes, which are involved in different kinds of imaging. And we have a kind of fake national node, which sits in our institute at LIACS, so we can demonstrate that this approach of the local and national nodes is a viable one. And this is what we're doing now. So this is the setup. We have information coming directly from the microscopes being ingested into the OMERO database. We have metadata from the users about the experiments being put into the OMERO database at the same time. You can't actually book time on the microscopes until you've done this bit. So we're enforcing the metadata capture part. Probably this is not going to be a popular move, but hopefully once people have done this a few times, they will see the value in that. As things are being analyzed and processed, sometimes they go outside of OMERO, so we have to capture more metadata about what's happened when it comes back in. In the future, of course, we want all of this to be within the OMERO database, so we want to be able to reuse successful workflows for analyzing imaging data, and we want to then be able to make those accessible to more people. Well, yeah, so a wider range of people, so people from the laboratory side as well as the image analysis side. So this is the general setup. There's a lot more to do to make it truly fair. So it's not completely finished. We need to standardize the way metadata is collected, standardize the way people describe their experiments. And for that, we can just simply reuse some of the technologies that we developed during the FAIRDOM days. So we can make metadata templates for data entry. We can adopt existing technologies for annotation from the community. We can develop our own where necessary. We have a piece of software called Writefield, which allows this kind of formalization of the metadata. And as Bern mentioned before, machine learning approaches are really rather a large thing. So being able to collect standardized metadata this way will enable an awful lot more automated metadata generation later on as well. I already mentioned the FAIR workflows. So this idea that these processes that people go through, a lot of them are common, and people are reinventing them all the time. We don't want that anymore. We want people to be able to identify from a publication, this is the actual image processing workflow I want to use. Here it is. I can just rerun it. And this will allow reproducible results, of course. And if we have this system, as well as making the workflows available, we also make the people who made the workflows available. It's easy to see who did what. It's easy to identify those people, so you can ask them to collaborate with you, and so on and so forth. And in the main proposal, we have a national help desk system that will formalize this process as well. And then finally, there's this piece up here. So how do we get information from our local OMERO to the national node, even if it's our fake national node at LIACS at the moment? And how do we manage the permissions for the people who are using the data? So OMERO already has quite a complicated system built in for sharing data. You can have private groups, you can have groups where you can just read and download data, you can have groups where you can add annotation, and you can have groups where everybody is allowed to read and write and change everything. Now, of course, when you're sharing in a fair way, then probably this is the way that you want to go. You have the data as it was published, as it were, and people want to download that, read it, understand what you've done. So this read group is basically what we have here in the public group. Okay, and so swapping between the OMERO instances is a challenge at the moment. There's nothing really to help us do that, so that's something we'll have to develop over the next few months. And I'll just finish with this summary then. So the bioimaging community, we can see that it's catching up with this idea of fair sharing and fair data, and the fact that the IDR is filling up with data as we speak is a testament to that. The OMERO database provides a flexible platform for building everything else on top of. It keeps researchers in charge of their data, so we're not breaking the rules that we set ourselves at the beginning. We're not reinventing, we're making sure that scientists remain in control. When we have the full system, what this will do is showcase Dutch results throughout the nation, but also throughout the European Euro-Bioimaging initiative, and it will clearly ensure that we do much better at reusable and reproducible data in this domain. Okay, now I'll leave it there. Thank you.
Thank you so much, Katie, and thank you so much, Bern, for your talks. And I would like to welcome Stuart Spallinger, who is the head of the department for Bern Space, who will agree to do the closing remarks. That's the wrong one.
This is the right mic? Okay, well perhaps you don't need it with so little people. So first of all I want to thank the two speakers who gave an excellent explanation of why open data and thinking about open data is useful and also a necessity. And I'd like to share with you briefly, because we're over time, a few thoughts and reflections on open data. So the first one has to do with, I would say, the application domain, because the field of microscopy and bioimaging is sort of partly overmingling with the medical domain, where we also use microscopy images and other images. And if you think about data in the medical domain, and especially about ownership and control of data, you're easily walking into an ethical minefield, because there the data is, I would say, in essence not open. The data belongs to patients, of which you, or at least of parts of which you take images. So that's something that's really important also to think about. Then about, let's say, the issue of metadata. One other thing I think is also interesting to consider or to reflect upon is the issue of compression. And I'm not talking about MPEG or any other codec. I'm talking about the notion that every bit that enters my PC that is not useful to me is garbage. It's bit pollution. And a lot of the raw image data that we are looking at, or that we're working with, is in essence containing bits that are actually no use for us. For example, these movies that we make to produce super resolution microscopy images, there are tens of gigabytes. In essence, they contain very little useful information, and only a set of coordinates of molecules that we localize in these images are the actually really relevant data. So that's also something to think about. Final step is that data always comes from data factories or microscopes or any other imaging instrumentation, and that's also important if you want to do comparative studies, to think about the instruments and the protocols that gave rise to this data. Standardization in that area too is also something that could be very useful to improve the value of open data. So those are just a few of the, let's say, few thoughts or reflections I have about this topic. And if you have time, I want to keep it with that. And if there are any questions to the speakers, then raise your hand, talk. Okay. Yeah.
I had a question about the metadata. I think this is maybe, this can be quite challenging, or I wonder that maybe, how do you foresee that? Sometimes I think it can be quite difficult. For example, if you report on laser powers in a metadata file, in my experience, when I put something in a commercial microscope, it's not the same as what I measure if I put a power meter, for example. And also the power can then next month be different again. So do you also have like procedures for how people should report these things?
Yeah, that's a good question. So, it's okay, I have this one. Yeah, so we want to ingest as much automated metadata as possible, because of course that always helps. But if you know that there are differences between the reported automated metadata and what you're actually observing, then that should definitely be a field in the minimum information model. That should be something that we capture there too. There's work going on in the community to develop a minimum information model for all microscopy imaging. So what's the least you should say about any experiment? As far as I know, that is not on the list at the moment, but it's an interesting point, and it will certainly something that we take back and talk to them about. Yeah.
I think especially, for example, if a data set takes intensity information to say something about stoichiometry of a molecule, for example, then that should be automatically be categorized as data that should contain a metadata file to show that how are the intensities actually derived or calibrated or so. So it should, I think it should be like somehow categorical that if your imaging data concerns this type of analysis, or if it is single molecule localization data, then it should have like a different set of metadata or maybe some controls for your microscope than another data set.
Yeah, so I said that there was work going on to develop the minimum information model, and that of course is quite misleading. For various different kinds of experiment, you would have the same basic minimum information model, but with extensions for various things. And I think this sounds like a very good example of why we would need that. So yeah, that's a very good point.
Thanks so much for the questions. I do realize that there are some more questions from the audience, but in the interest of time and to make sure that whoever is in a rush home to their families can leave the room without disturbing everybody, I would like to conclude the event, but I would like to thank both of the speakers again and Stuart for doing the wonderful closing remarks. So please join me in thanking all of them. And as always, I would like to thank everybody who was involved in organizing this event. There was lots of colleagues who collaborated on making this happen, so thanks to all of them, and thank all of you for coming. And hopefully our speakers will be able to stay for a drink or two to answer all the questions that I'm sure there were in the room. And the final thing, which I think is a good announcement, my colleague Yen, who you can see there, maybe you can stand up, Yen, will lead you to a room where we have a dedicated breakout drinks and little nibbles, so hopefully we can continue discussing the questions and how to make microscopy data fair in the other room. So please, let's follow Yen. Thanks again.